Good morning, everyone, or afternoon for some of you. I want to thank everyone for attending the webinar this morning on our devTOX quickPredict assay. My name is Jessica Palmer, and I'm the Associate Director of Toxicology at Stemina Biomarker Discovery. And I just wanted to let everyone know, if you have questions, please type them into the question and answer box, and I will try and answer everything live, assuming there's time at the end of the presentation. So a little bit of background on Stemina to start with. Stemina was established in 2007, and we opened our laboratory in Madison, Wisconsin, in the University Research Park there. And it was designed really to have state-of-the-art mass spectrometry and stem cell culture capabilities. Our expertise really focuses on human stem cell models of toxicity and disease. And over the years, we've really developed a unique and proprietary metabolomics platform. Our sister company, NeuroPoint DX, which is the part of Stemina that focuses on diagnostics, right now is focused on developing a diagnostic test for autism spectrum disorders. And really the focus and goal of Stemina has always been to identify biomarkers of toxicity and disease for the diagnosis and more precise treatment, drug development, clinical study management, and companion diagnostics. But today, really, I'm going to focus on the toxicology side of our business. The approach that we took at Stemina was we decided to build upon the human pluripotent stem cells that were really developed and identified at the University of Wisconsin-Madison. And we saw them as a unique opportunity to evaluate chemical toxicity using a human endpoint, which really allows, using the human endpoint really allows elimination of species-specific differences in toxic response. These cells can be differentiated into cells that function very similar to their in vivo counterparts. They have the ability to self-renew in culture, so you can really have an unlimited supply of them. And they're applicable to high throughput systems. We use embryonic or induced pluripotent stem cells in their undifferentiated state for their devTOX quickPredict assay. And then we also use induced pluripotent stem cell derived cardiomyocytes for our cardio quickPredict assay. We've combined the human pluripotent stem cell biology with LC-MS-based metabolomics. And we chose metabolomics because really the metabolome is considered to be the closest to the phenotype, and it can be impacted by any of the previous components of the flow of information. So changes in gene expression, changes in transcripts, and changes in proteins can all lead to changes in metabolites. And so we've used these two we've used the human pluripotent stem cell biology and LC-MS-based metabolomics to develop our in vitro toxicology assays, with the goal of developing assays that allow our clients to understand compound risk profiles faster, looking at multiple exposure levels, and really with a minimal amount of compound required. We only ask for 15 milligrams of compound for the assays when we conduct them. And the goal is to really provide a cost savings compared to the standard in vivo tests that need to be done for developmental toxicity or cardiotoxicity. The two assays, as I mentioned, that we've developed, will really provide an exposure-based prediction of toxicity potential using targeted metabolomics. So we started with an untargeted platform and then migrated it to a targeted platform for the final assays that we deliver. Today, we're going to focus on the devTOX quickPredict assay. And if you or any of your colleagues are interested, I gave a webinar last week on our cardio quickPredict assay, which should be available soon on our YouTube channel. So the assay was initially developed using human embryonic stem cells, which are derived from the inner cell mass of a blastocyst. And as I mentioned, they are pluripotent, meaning they can differentiate into multiple cell types and have the ability to self-renew in culture. We do not derive stem cells in our lab. What we do is we use lines that are already well established and well published. And so for the embryonic stem cells, we use the WA09 line that was derived by James Thompson in 1998. When human-induced pluripotent stem cells were identified and methods became reproducible, we chose to optimize the assay for use with these cells. And so induced pluripotent stem cells are derived by taking a somatic cell and basically de-differentiated it back to that pluripotent state. And these cells, again, have the same capabilities and abilities that the embryonic stem cells have. To identify the biomarkers in the devTOX quickPredict assay, we exposed the cells to a training set of 23 well-characterized compounds that had known human developmental toxicity outcomes. So these are largely pharmaceuticals that we used early on in development that had good human epidemiological data, either showing that they were developmentally toxic in humans or not developmentally toxic in humans. And we did multiple replicates of untargeted metabolomics to look at changes in the secreted and consumed metabolites. So we're not focused on measuring the metabolites within the cell, but measuring how those cells are interacting with their media. And as a way of understanding the cellular biology, which allows us to use the cells for other downstream applications. Doing this, we were able to identify we could develop multiple different complex models for predicting developmental toxicity. But when we looked at could we narrow that down to specific metabolites that were key for predicting developmental toxicity, we identified 2 metabolites that were able to predict the potential for toxicity just as well as those more complex models that included many unknown features. So these 2 metabolites were ornithine and cystine, and I'll go into their biology and why they may be important for embryonic development. So ornithine is secreted by the cells into the media, and changes in ornithine most often will decrease with toxicity. And so and often when it decreases, this is indicative of cell death, but there are also mechanisms that could cause a decrease in ornithine where this independent of changes in cell death. There are instances where you see an increase with toxicity. The most common, or the one that we've seen most often, are compounds that are related to the retinoids and all-trans retinoic acid, where you'll see this increase with higher concentration, and that's due to the impact of retinoids on ornithine decarboxylase and their ability to inhibit ornithine decarboxylase. Metabolically, they are part of the urea cycle and one of the rate-limiting metabolites for polyamine synthesis. And so they can impact, changes in ornithine could impact DNA stability. In contrast to ornithine, cystine is a media component, and it's consumed by the cells from the media. When the cells are happy and healthy, they are using about 75% of the cystine that's in the media. But when you treat them with a developmental toxicant, you'll see an increase in toxicity, indicating that the cells stop consuming cystine and stop taking it intracellularly. And this can occur through multiple mechanisms. Metabolically and biochemically, it's involved in cystine's main function in the cell is to help regulate oxidative stress. And a decreased cystine uptake likely reflects a change in cellular glutathione synthesis and the redox balance. Both of these metabolites are essential for normal proliferation and differentiation during development. And so you can imagine if either of these are disrupted during early embryonic development, that could lead to malformations or embryo lethality later on. So the process that we use is as I mentioned, we culture the cells in their undifferentiated state. For IPS cells, we use a line that's commercially available through ATCC. And we'll plate them into 96 well plates with 2 chemicals on each plate, looking at 8 exposures of every compound. In addition, every plate will contain reference controls and positive and negative controls to ensure that the assay is performing within the specifications of development. And this also helps to confirm that the cells are maintaining their they're karyotypically normal. We'll expose the cells to compound for 48 or 72 hours, depending on the cell line. And then we'll collect that spent media for LC-MS analysis. And we'll use a targeted approach to measure ornithine and cystine, specifically, as well as internal standards to help account for any variation that may occur due to LC-MS measurements. Because we're using the spent media, this leaves the cells, as I mentioned, for other downstream analyses. And with every metabolomics assay we perform, we also do a cell viability assay to look at are the chemicals impacting the viability of the cells. And then we'll do data normalization and dose response modeling to understand the changes in the ornithine and cystine as well as in cell viability. And so if you look at that graph a little closer, the assay really predicts potentially toxic exposures and is built around the idea that toxicity is a function of exposure. And so to associate you with the graph here, you have your test article concentration along the x-axis and then changes in viability or the OC ratio along the y that have been normalized to the DMSO controls. And so using that training set, we identified a level of exposure that was indicative of developmental toxicity. So how much change in the OC ratio was required before a compound would be predicted developmentally toxic. And then we can use that to identify at what point does this ratio cross that threshold and what concentration would be predicted to be developmentally toxic. That data can then be compared to cell viability to understand mechanisms and whether or not the ratio is changing independent of changes in cell viability or if changes in the ratio are reflective of changes in viability. And using this data, we can determine non-developmentally toxic and developmentally toxic exposure windows. So there's 3 main types of response that we observe with the assay. And the first one is really the simplest, where you don't see a response in the ornithine or cystine or cell viability. And these compounds would be predicted to have little to no potential for developmental toxicity in vivo at the exposure levels we're testing. The second is when you see changes in the OC ratio independent of changes in cell viability. So this could manifest as no change in cell viability, where you see a decrease in the OC ratio, such as we're observing here for carbamazepine, which is largely driven by this cystine response. Or you could see a viability response that's occurring at concentrations significantly higher than concentrations in cell viability. And we use a threshold of 3-fold difference between these 2 changes, or between these 2 concentrations, such as we're observing here for busulfan. And these compounds we predict to have potential for developmental toxicity in vivo at or above this DTP concentration. The third response we observe is really the one that causes is the most difficult to interpret. And that's when you see a response in the ratio and viability at similar concentrations. And as you can see here with aminopterin, which is a known human developmental toxicant. And so when you see this, if there is no other additional information available, we use really a public safety type of prediction to say it does have potential for developmental toxicity and or embryo lethality in vivo at or above these concentrations. And this is because you can imagine if you're killing these cells, you would be, because they are directly reflective of the embryo during those early stages, you're likely to kill that early embryo. However, if there is additional data available, you could use that to help interpret this and determine whether or not this response in cell viability is likely to be specific to the embryo or if it's likely to be reflective of a non-specific cytotoxic response. For example, if there's data from additional cell types such as fibroblasts, keratinocytes that show a change in cell viability at similar concentrations or lower concentrations than we were seeing a response in the OC ratio, that would be indicative of a non-specific response. And the prediction for developmental toxicity would be qualified with that. So to understand the predictivity of the assay, we've tested a very diverse chemical set with pharmaceuticals, agrochemicals, cosmetics, and environmental chemicals. And we've compared this data to published human and in vivo results. And for every compound that we test internally, we will identify all of the rat and rabbit low-L and no-L data, and if there's corresponding Cmax values available, that can be used for prediction. And we're working on a joint publication right now as part of a Tox21 cross-partner project that we have completed with NICEDM, the EPA, FDA, and NCATS that will describe the compounds that we tested for them, as well as the larger compound set that have been tested at Stemina, and include in vitro to in vivo extrapolation to help understand how the DTP concentrations we're predicting reflects in vivo doses. And so we use two different prediction models based on the available data. So if we can find exposure data, whether it's human for compounds that are known to cause developmental toxicity in humans, or rodents for the Cmax for different low L's, we'll score that DTP against that data. So if you have a DTP concentration that is above 10 times the Cmax, that compound would be predicted to be non-developmentally toxic. If your DTP concentration is below that, 10 times the Cmax, the compound's predicted to be developmentally toxic. And as I mentioned, this is really just a way to understand the performance of the assay. And the in vivo data is not necessary for interpreting the results, but it does help put them into context with the in vivo situation. When we don't have exposure data, we score the DTP against a concentration threshold that was again developed using that training set to balance accuracy based on balancing sensitivity and specificity. So if your DTP is greater than 65 micromolar, we predict that compound to be non-developmentally toxic. When the DTP is less than 65 micromolar, we predict it to be developmentally toxic. And using these two models, you can see that the assays really perform quite well. And with HES cells, the data that I'm presenting here is for 79 chemicals. And for IPS cells, the data is for 124. And based on these chemical sets, we're able to obtain a balanced accuracy of 83% for HES cells and 87% for IPS cells, with sensitivities of 88 to 90% and a little bit lower specificity in HES cells. So we have a few more false positives but excellent specificity in iPS cells with 86%. And there are differences in the compounds between these two. So this difference in specificity doesn't reflect differences in prediction between the two cell types, but differences in the compounds that were tested. And if you look at this data and narrow it down to understand how is the assay performing when you look at just human data, there were 35 compounds in the set that had good evidence of human developmental toxicity. And when you compare how the assay is concordant with the rodent system, we were 74% concordant with how the rodent predicted the human outcome. And the rodent model predicted human outcome with 86% accuracy, and we predicted the human outcome with 89% accuracy. Compared to the rabbit, 28 of the compounds were tested in rabbits. We were 79% concordant with the rabbit data, with the rabbit predicting human outcome with 79% accuracy, and we predicted the human outcome with 86% accuracy. Now, looking at the predictivity of the assay compared to other commonly used ex vivo and in vitro assays, I was able to find data and pull data from the literature for about 25 compounds for each of these assays. Our concordance with these assays ranged from 65 to 75%, and that lower concordance is specifically due to a difference in the specificity of these assays. So there were these assays had more false positives than the devTOX quickPredict assay did. And so when you look at the performance of those assays compared to the human outcome, they were about 75% accurate at predicting human response, whereas our assay was greater than 90% for the same compound set. So the main focus of this webinar was really to talk about the reproducibility and the applicability domain of the assay. And so reproducibility, we've measured in a number of different ways and tracked over time to make sure that the assay continues to perform with the same accuracy as it was developed with. And so every time, one way that we assess accurate reproducibility is to look at a set of 2 to 3 compounds or drugs to check metabolism in every new batch of cells that we thaw. So every time we bring new cells into culture, we'll pre-qualify them with carbamazepine, methotrexate, and thalidomide, and in order to make sure that the cells are responding, not only showing a response, but they're responding at similar concentrations to where that response was observed with the previous batches. And so you can see, the goal is that we want all of our predictions to be, or all of our DTP concentrations to be within 3-fold of this mean value. And with the exception of one replicate in carbamazepine, over the last 6 years, 39 of 40 batches have passed that and been within 3-fold of this mean. For methotrexate, you can see the range is a little bit smaller, and all 40 of those have been within 3-fold of the mean. And thalidomide, we recently added to our testing and qualification, and again, you can see the reproducibility over the last 3 years has been excellent, showing very little variation with that DTP concentration. And so most often, the slight changes you see in the DTP concentration aren't from differences in cellular response, but differences in where that dose response curve is crossing the threshold based on slight, between 2 concentrations, I should say. And so in order to maintain and ensure assay reproducibility, we continue to do this every few weeks. We also can look at reproducibility using the plate controls that I mentioned. And so we want to ensure that every plate and every experiment we do contain the cells are metabolizing ornithine and cystine, and any predictions that we give are going to be consistent with assay specifications. And so looking at inter and intraplate variation over the last 6 years, this data is for 3 years from 2016 to 2019, the mean intraplate CV was 4.1%. So within a plate, within the positive and negative controls, the CV was 4.1%. When you look at the data from all of the plates and the reference, the negative and the positive controls, you can see that across plates, again, we have a mean CV of 9.9%, with a little bit higher in the positive due to magnitude differences in the OC ratio response and due to magnitude differences in the cystine response. But as you can see with the data on the previous slide, this magnitude of change doesn't affect the ability or doesn't affect that concentration where a DTP or where the OC ratio crosses the threshold. And so moving into the applicability domain, you can see the assay is really, there's a number of ways you can look at the data and define the applicability domain of an assay. And so the one is to look across chemical classes or industries. And so if you look at the different industries, pharmaceuticals, agrochemicals, industrial, personal care products, you can see that the assay performs with excellent balanced accuracy, regardless of the industry or the product type, with high sensitivity and high specificity. You can also look at the applicability domain to understand, are there specific types of birth defects that are not predicted well with the assay? And so while the assay does not predict the type of birth defect a compound is going to cause, it tells you whether or not it's going to cause a birth defect for any of these classes. And again, it has excellent accuracy across the different birth defect categories, with the lowest being growth restriction, which is more of a developmental effect instead of a teratologic effect. But when you look at the main teratological effects, the lowest would be something like gastrointestinal or eye, which would kind of make sense considering the early endpoint that we're using and the early point of the cells. When you get down into pharmacological mechanisms and the targets of the assay, there are a number of ways that you can parse the data. And so I'm going to present data from looking at pharmaceuticals and then data from the recent publication by the EPA. And so you can, in the recent update to the ICH guidelines, they provided a list of compounds and a list of pharmacological classes to help understand the accuracy of alternative assays that may be used for drug development. And when you parse the data for all of the pharmaceuticals that we tested, which includes a combination of the ICH reference compounds and additional pharmaceuticals tested at Stemina, you can see across the different pharmacological classes, the assay performs really well with high sensitivity, with the exception of receptor modulators, and where your sensitivity drops to about 50%. And so I'm not going to show a lot of the different reasons for this, or a lot of different graphs that demonstrate this, but what I will expand upon with this is to say, when you dive into those receptor modulators that the assay is not predicting, what I found was the receptor, the assay is not able to predict chemicals where the receptors are not expressed in human pluripotent stem cells. And so it's really important to understand what the capabilities of your assay are and use all your background data to help interpret your results. And so an example of a receptor modulator that I thought is important and interesting to show would be fingolimod. Now, this compound, when we initially tested fingolimod, you can see that it's causing a response in the OC ratio, but not until you get up to concentrations that are more than 10-fold higher than where this response was observed in rodents and 100-fold higher than where the response was observed in rabbits. And so looking into the data, you can find that fingolimod actually has to be metabolized into its active metabolite. And so it's not the parent compound that's interacting with the receptor in vivo, it's this active metabolite. And the cells do not express the kinase necessary to convert fingolimod into its metabolite, fingolimod phosphate. And so when we looked at the metabolite, you do see a response in the cells at concentrations that are within the levels where you saw a response in vivo. And there were without any changes in cell viability. And looking at the receptors that this compound, this drug, interacts with, you can see that the HES cells do express one of the sphingosine receptors, which could be the mechanism whereby we're seeing an effect in the OC ratio. But what's also important to point out is that while we don't see a response at the concentrations in vivo with the parent compound indicative of interactions with the receptor, we do see a response that could be indicative of off-target effects. And so my point with this compound is really twofold. One is that, the cells have very limited metabolic capabilities, and while I'm always looking for ways that we could induce metabolic capacity with the cells and looking at exogenous metabolism methods. Right now, there hasn't been a good, there hasn't been one that is available that works with the cells without killing them or interacting with cystine and ornithine metabolism. And so whenever possible, it's important to test both the parent and the active metabolite when you know that's there. The second is to understand, the second point of this example is to understand the expression of different receptors and kinases within the cell and understand whether or not your biological system is expressing the target of interest. So the EPA ToxCast, we had an EPA ToxCast contract and did a number of studies for the EPA over 5 years. And the culmination of all of that data was recently published in Toxicological Sciences. And so this study was conducted with human embryonic stem cells. Most of the data that I had been talking about was with iPS cells previously. But really, like I mentioned, there's no difference between the two cell lines. And so the way that the study was conducted is we tested a total of 1,065 compounds in 2 different types of assays. 686 of them were tested at a single concentration, and 379 of them were tested with the devTOX quickPredict assay in an 8-point dose response curve. And so these 1,065 compounds were then processed using the ToxCast data set, using the ToxCast data pipeline, and that data was interpreted. And then once they went through all of that, went through that entire pipeline, that data was is deposited in the in vitro database available from the EPA. And so the major study findings from this large study was that 19% of the chemicals were predicted to have developmental toxicity. And the assay performance compared with in vivo animal models of human response varied from 79 to 82% accuracy with higher than 84% specificity but about less than equal to 67% sensitivity, which is significantly lower than what we had seen with the chemicals we've tested in-house. But what they found was that sensitivity really improves as more stringent criteria are applied to the weights of evidence. So basically, if you have more confidence in your animal study, the assay has a higher sensitivity and accuracy, which would make sense because the compounds that you don't have as much confidence in, such as they were predicted to be toxic in one but not predicted in another species, are ones that you wouldn't be as confident as whether or not they were going to be human developmentally toxic. And they took this data and they compared it to the biochemical targets in the ToxCast set of in vitro assays to really understand positive and negative associations with the response observed with the devTOX quickPredict assay. And that data can be used to provide insight into the mechanistic underpinnings of the targeted endpoint and its biological domain. And this is really what I want to focus on in the next slide and discuss. So to understand the applicability domain with the ToxCast data set, what the EPA did was they mined statistical correlations for the response in the devTOX quickPredict assay with a set of enzymatic and receptor signaling assays, which is the NovaScreen data set. And they looked to see, and then they did a number of different steps to narrow that down to the most biologically important pathways. And so, removing different genes that would not necessarily be important for developmental toxicity or important in human response. And from this, you can really identify which pathways fall into the Stemina positive and the Stemina negative domains, or which molecular initiating events in different pathways fall into those. And what they found was that in the sensitive domain, really it was focused around receptor tyrosine kinases and associated downstream kinases and phosphatases that are important for cell growth, differentiation, and survival. And in the insensitive domain, the genes that were at the top of the group were calcium second messenger generation, or GPCR signaling, and nuclear receptor mediated gene expression, with the exception of NR3C1. So the assay for chemicals that were believed to be acting and causing developmental toxicity through these pathways, the assay was not responding to them. And when you look into those different genes, you find that many of them are not expressed by human pluripotent stem cells. And really, these results are consistent with the applicability domain defined using the pharmacological class approach, where you're seeing kinases and enzymes showing a positive response in the assay, and receptors, especially receptors that are not expressed in the cells, showing a negative response in the assay. And so with that, I'll wrap up and take questions. But in conclusion, the devTOX quickPredict assay is really an accurate and selective assay for predicting human developmental toxicity. And really that is, the key goal of anything that we do. That being said, when you look at the prediction of devTOX compared to rodent or rabbit in vivo predictions, the assay is about 80% concordant with rodents or rabbits. So even though our goal is to predict human response, we are able to predict what your in vivo tests will likely say with excellent concordance as well. The assay can be reproducibly measure the dose response over time using multiple cell lines, consumables, reagent lots, and technicians. So you can be confident that a result that you get now is going to match a result that you get 2 years from now. And it has the ability to identify a wide range of mechanisms, but we do recognize it may not be applicable in all cases, such as when targets are not expressed in the cells. And even when the targets are, there are cases where those specific targets of a compound may not impact ornithine and cystine metabolism in the end. But it is important to also point out that when the targets aren't expressed, the assay could still provide useful information and evidence of off-target effects that may impact development. And so what's next is our goal is to incorporate in vitro to in vivo extrapolation, which is something we're currently working on with Nicedum, and I'm hoping to provide this as an outcome in the report that is provided to clients, as well as the DTP concentration down the line, as we work to incorporate this into our predictions. And in addition, the EPA is working on a machine learning approach that develops predictive models using the devTOX quickPredict assay and other in vitro and in silico models to provide a more comprehensive prediction of developmental toxicity. And so what they're doing is looking at those areas where the assay, those areas that fall into the assay's insensitive domain, and trying to understand what assays could be used to fill that in, and would complement the prediction being provided by the devTOX data. So with that, I want to thank everyone for attending today, and I will I'll work through any of the questions that are available, but I want to thank my team at Stemina. That is, without them, none of these results would have been possible. And to let everyone know, we are now open and performing work now for our clients. And so if anybody has chemicals that they're interested in testing, please reach out to me. This is my contact information. And we can set up a time to talk further and hopefully conduct studies. So with that, I will move to the questions. So the first question is, did you observe differences in tox in the stem cells and differentiated cells like cardiomyocytes? And so the answer to that is yes. When you look at the biomarkers that are important for the devTOX assay compared to the biomarkers that are important for predicting cardiotoxicity, you can see that they do not overlap. And so, we looked at drugs that, when you look at the data set that we've tested, a number of the non-developmental toxicants that we've tested are known to be toxic to other systems, such as cardiomyocytes or hepatotoxicity. And you can see they're not causing a response in ornithine or cystine. When you look at those cells in the other cell types, the biomarkers that we use for quickPredict are lactic acid, arachidonic acid, 2-deoxycytidine, and thymidine. So very different mechanisms that are important there for predicting cardiotoxic response. And so, yeah, we did look at that, and there are differences. And so, we're not just predicting a general development or a general toxic response with the embryonic stem cells. It really is specific to developmental toxicity. Are there other questions that people have? If not, if you think of something later on, please feel free to contact me. And I will answer everything to the best of my ability. We will be following up with a copy of this presentation, which does include links to all of our most recent publications. And if you are interested and need a copy of the recent publication with the EPA, let me know and I can send that to you as well. With that, I'd like to thank everyone for their time this morning, and I hope everyone has a great day and a great weekend